Hi, everybody. This is our lesson 8 on ordinations. My name is Liz Suter. I am an assistant professor of environmental sciences. And so we'll jump right into this lesson, which is posted both on the BVCN GitHub and on RStudio Cloud. So to start, we need to install some packages. This tutorial mostly uses a package called PhyloSeq for all of the analyses that I'll be demonstrating here, which is also dependent on a lot of the plotting functions in tidyverse. And we'll also use a package called ape, which is for manipulating phylogenetic trees. And the first step after that would be to import data. So I'm using data here that I worked with in lesson 4 of Amplicons, in which I cleaned up some data that was the result of a QIIME pipeline. So QIIME took our FASTQ files and we used it to make OTU tables and taxonomy files. And then in lesson 4, I used that output to do some manipulation to just normalize data, clean it up a little bit, remove some, detect contamination, remove that contamination, and so on. So that is what we're importing here. And then in this code block here, I'm importing some metadata. So the metadata are directly from GitHub. The original source of these data are the same data that are used in the Happy Valley tutorial that we keep mentioning in Amplicons. So the metadata just indicate what the different parameters are for the different samples. So I will run both of those code blocks and you'll see all of my data appeared in my environment here. So I have a FASTA file, a count table, sample info table, which is the metadata, a taxonomy table, and a tree file. So before doing ordinations, there are a few more sort of cleanup things that we have to do in order to make them compatible with PhyloSeq. So the first one here is formatting the taxonomy table. So if we take a peek at the taxonomy table using the function head, you can see that there are, so here are the feature IDs. So these are sort of the unique IDs of the ASVs that QIIME gave to my ASVs. And then if you look at the actual taxonomy, this is a character vector with some weird letters in between the different taxa, right? So it's indicating like the first level is bacteria, and that is preceded by a D_0 and what looks like 2 underscores, and then a semicolon, and then another D, and so on. So what I want to do to this table in order to make it compatible with PhyloSeq is divide each of these taxonomic levels into columns. So that's what I'm doing here using 2 major functions, mutate and separate. These are from dplyr, I believe, in the tidyverse. So I'm going to make a new data file called taxonomy, which takes my taxonomy no-contan, which I just imported, and takes that into the function mutate. And within mutates, I'm looking for a string, which I'm going to replace. And that string has this pattern, D_, and then this is called a regular expression. And I put a link to a cheat sheet here. So regular expressions are these sort of codes that indicate that the expression that I'm looking for can look anything like a certain pattern. So in this case, I'm telling it it's surrounded by an underscore and there's a digit. So it will be D_digit. And I used just a generic digit instead of telling it 0 or 1, because if you look at my taxonomy table, there's zeros here, there's ones here, there's 2s here, 3, 4, 5, and so on. So I wanted to replace that in every case. And that's the sort of cheat to do that. I'm also looking in the next line to replace those semicolons, which it will remove. And then I start to separate the taxa into columns. So it separates taxonomy, and I'm naming my columns kingdom, phylum, class, and so on. And then I'm going to continue on. I'm going to remove taxon, the column. So up until this point, I'm making new columns based on what was in my taxon column. After those are made, I can remove the taxon column by using select and put a negative sign in front of taxon. And I'm also removing a column called confidence, which I didn't talk about, but that was, so that's some important statistical measure of how confident we are that each of those ASVs belongs in that taxon. So I'm removing that in order to make it compatible with phyloseq. And I'm also telling it to use the feature ID column as the row names. So let's run this. And now we can take a peek at it down here, again using head. And this is what it looks like. So that's a pretty straightforward taxonomy table, and this is the format that's expected by phyloseq. And you can see kingdom, phylum, and so on, keeps going. So our taxonomy table is ready. So some more formatting for a phyloseq package is to take the count table, the taxonomy table, and the metadata table and convert it into forms that phyloseq expects. And this is done by using functions from phyloseq called OTU table, tax table, and sample data. So I'm literally just taking my count table, using the function OTU table, and making a new data variable which phyloseq wants. Same thing with my taxonomy. I'm using the tax table, and with my sample info table, I am using sample data in order to make that into a variable called meta, which is a phyloseq object. I should also mention that in the last tutorial, I already used phyloseq to import my tree, so it's already a phyloseq object. So run this, and then I just want to check that phyloseq will know that what are the taxon names and what are the sample names in those. So phyloseq has a function called taxon names and sample names. So I want to make sure that I imported those correctly so that when I say what are the taxon names of tax, it'll tell me the correct thing. And we can just peek at those. And these look like the taxon names that Chime gave to my ASVs. So that looks correct. And using sample names, those are also the names of my samples for both the ASV object and the meta object. So that looks good. So I have these 4 objects, the ASV, the tax, the meta, and the tree, which I made last time in the previous tutorial. So now I can finally turn these into phyloseq objects. I'm just going to call the phyloseq object PS and run that. And it appears here as a large phyloseq object. So phyloseq makes your life really simple when you're using Amplicon data. So before I started using phyloseq, I would use a lot of functions in tidyverse to sort of manipulate data into the way that I wanted to like view it in a plot, or to transform it in a way that I wanted to do statistics on it. What's neat about phyloseq is that a lot of those like underlying manipulation steps are built into the functions themselves, so you don't have to worry about all those extra lines of code and doing that. So some of the functions, for example, of phyloseq, besides the ones that we already took a look at, are things like rank names. So using things like rank names and tax table, we can just take a peek of what these data look like. So I run this. It detected the rank names, which I associated in a previous step with the taxonomy table, right? So it knows that kingdom, phylum, and so on is there. Then I told it to take a look at the unique kingdoms. And we have archaea and bacteria, which we expect, but we also have some things that we don't expect. We have an unassigned kingdom category, and we have the eukaryotes also. And then I said, well, give me a table of how many of each of those there are. And it is mostly archaea and bacteria, but there are 6 eukaryotes and 2 unassigned. So since this was a data set that used 16S primers, there essentially should not be eukaryotes. This is an error. So I want to remove those, and I want to remove the unassigned annotations. So I can filter those out using another phyloseq function called subset. So I'm saying subset from my PS table anything that has a kingdom that is unassigned or eukaryotes. And again, give me the sum within each kingdom, and now it's just showing the archaea and bacteria. You can do some similar things with the phyla. So we can take a look at the unique phyla names that are remaining in the table. And so there's a lot of expected things here. For example, you see Nitrospira, Planktomycetes, there's a lot of proteobacteria. So these are things we expect, but we also have some that don't necessarily have a phyla assigned. And then we also have some that where we have one ASV from the entire data set in the phylum. So for example, here's one in the epsilon proteobacteria, here's one in here, there's another one here. So these really low abundance phyla, you can try to remove them. I actually, when I was putting this together, I tried doing the analysis with and without those low abundance, and it didn't make a big difference. But sometimes those rare taxa can alter your interpretation later on a little bit. So in case you want to remove those or remove ones that were not assigned, you can do it in a very similar way that I did it for kingdom. So subset taxa that have a phylum that is in any of this list that I made based on looking at this table. So I'll run this. And now you can see our unique phyla that are remaining are the same except for those ones that I removed. So the next step is something that I included that's important only if you want to do a unifrac PCOA later on. And I'll explain what that is in a little bit. But a unifrac analysis depends on phylogenetic relatedness, which it gets based on the tree object within the phyloseq object. So I just took off a bunch of ASVs from my phyloseq object. It's possible that one of those taxa were actually the root of my tree. So if I just took those off, I messed up my tree file. So you can actually reroute the tree, which will help unifrac later on. And then there's also a step here that I've also learned from experience that you have to change the format of the tree if you use TRIME into this specific thing called the dichotomy tree. So I put a couple links here that where I found helpful for figuring this out. So here I'm actually defining a function that looks at the tree file and is trying to pick out the branch that's the longest, and it's assigning that as the outgroup of the tree, and then I'm rerouting it based on that outgroup, and then reassigning my tree to the phyloseq object. So I will run this just so we have it. And in the last step here, it's assigning that new tree to my PS object, so I know that it's still there. Okay, so that will help us in a few steps in a little bit. But before I actually jump into the ordination, sometimes you might want to take a look at the distribution of taxa in your data set. So I think we can probably teach an entire lesson on plotting Amplicon results, but I just wanted to take a quick peek here just to make sure that everything was looking like it was supposed to and looks similar to the happy belly analysis, for example. So another really neat thing that phyloseq does is it has this function called taxglom, which groups at a certain level. So I'm telling it to agglomerate my data at the phylum level, and I'm calling that phylum gloms. And then I just plot it with another phyloseq function called plot bar, which makes a bar plot of my agglomerated data. And so you can see here, each sample going across this axis and the abundance on this axis. So this, in case you didn't see lesson 4, my abundance here is already transformed. So it's a small decimal number instead of absolute numbers. So if you did this with an OTU table that was absolute numbers, it would obviously be different. But it still gives you a quick peek at just how the data look. So you can see like proteobacteria are the most abundant in each case, which makes sense, and which was also the case in happy belly. There's also a lot of tau marchiota here, for example. So things that you would expect. So that's looking good. Okay, so our phyloseq object is ready now for ordinations. And I'm going to cover 3 versions of an ordination here. the first is a PCOA, but before I jump into the PCOA, which I put a little bit of text here about what a dissimilarity matrix is. So ordinations depend on an input data set that is usually often we use something called Bray-Curtis dissimilarity. So this is a calculation of how dissimilar each sample is from every other sample based on the distribution of ASVs within that sample. And actually, if you go to the help menu and you look up this distance function, or if you look it up in vegan, I think it's called VegDist, it actually shows you the actual algebra that it's doing to figure this out. But anyway, if you take a look at a distance matrix, it looks something like this. So we have the samples going across as both rows and columns. And each number in here is telling you how dissimilar each sample is from every other sample, right? So the diagonal of this should actually be 0, I guess, because if you're comparing one sample against itself, it should not be dissimilar at all. It should be similar. And then respectively, every other sample should have something between 0 and 1. So these matrices also, they're usually empty on one half of the diagonal, right? Because that would just be repetitive. So when you compare BW1 to BW2, you don't need to compare BW2 to BW1. It would be the same exact number. So that's what a distance matrix would look like. And you can look at it on its own, like I did here. But when you actually do the PCOA, you don't even have to do this calculation up here of the distance matrix, because within the function ordinate in Phyloseq, you can tell it the distance method that you want to use. So here I'm telling it to use the Bray method. I'm also doing a PCOA, so my method for the ordination is a PCOA. And it's a really straightforward step. You just use a function ordinate to do this complex matrix algebra. So what the PCOA is actually doing is, so the distance matrix is a multi-dimensional matrix, right? It has many dimensions here. So a PCOA essentially takes the projection of that dissimilarity matrix and tries to rotate the coordinates. So keeping the data set distances the same, it rotates the coordinates around the data set to try to maximize the amount of variability explained by each axis, so that your first two axes explain the most variability. So those axes are essentially eigenvectors, and each of those axes is associated with an eigenvalue, which is a scalar number, which tells you how much relative variability is explained by that respective axis. So the step to actually run the ordination and run the plot is really straightforward. But understanding it is a little more complicated, right? So here I'm plotting the first two axes. And I also color-coded and shape-coded the plots for, or the points for each sample type. So they are color-coded by, I guess, the type of sample the type of rock they are, and the shapes correspond to the type of sample. So we have rocks, water, and biofilm. And within the rock category, there's altered, carbonate, and glassy. So you can see, based on the distance between these points, how they're separating. And remember, the separation is based on the dissimilarity between samples or the distribution of ASVs. So we have like the altered rocks here, the glassy rocks here, there's a carbonate rock here, and then water and biofilm are out here, and they look very different than the rock samples. One thing about this PCOA, though, is that the axes don't explain that much variability. So collectively, axis 1 and 2 is like about 30% of the variability. Oh, I added it here, 29.9%. So that's decent. I don't know if Sarah or Ella want to chime in on how good that is, but sometimes you can get much better numbers than that. So one neat trick that I wanted to add in here is that so the eigenvalues associated with each axis is different. So the way that I plotted this plot here, it's essentially a square. But each axis is associated with a different amount of variability. Here it's not too different, 13% and 17% is not that different. But suppose you had an axis that represented 50% of the variability and the other one was only 15%. Representing those two axes as the same length is not really fair when one corresponds to a greater amount of the variability. So you can actually scale the plot based on the eigenvalue that each axis is associated with. And that's what I'm doing here. So I'm essentially plotting the same thing, but I'm calling it the PCOA plot scaled. And I'm scaling it by a value that I called evals, and that is a calculation based on the eigenvalue from axis 1 and axis 2. So this is not going to look too different because, again, this is 13% and 16%, but I got this idea from the Bioconductor website, and they have an example where one is much longer, and so this really stretches out the plot and indicates that axis 1 explains much more of the variability and therefore much more of the differences among the samples. Okay, the next type of ordination here is a weighted unifrac, PCOA. So in this case, I'm still doing a PCOA, but I'm using as a distance matrix the weighted unifrac method. And this essentially takes into account how many taxa from each respective pair of samples are related to each other. So accounting for that phylogenetic relatedness will change the interpretation a little bit. So, but the code is very similar. So we still use the ordinate function and we still use the PCOA method, but here we're just using a different distance metric. And if we look at that, what's pretty neat about this is now we still have a clear separation of the water samples and the biofilm sample from the rock samples, which are these three colors up here. But now the altered and the carbonate are overlapping. So to me, this is saying that there are different ASVs in these samples according to the distance matrix, but according to the unifrac matrix, the ones in the altered and the carbonate might be phylogenetically related. So sometimes it's worth it to do multiple ordinations. You learn a lot of things about your data. And I also put a note here that, well, first of all, this ordination explained a lot more of the variability. So using this distance matrix actually counts for about 34% and 24% in the first and second axes. So this is over 60% of the variability. And if you were interested, for example, in what ASVs were accounting for the difference up here, that is not accounting for the difference up here, you could do some downstream analyses, which maybe we could talk about in another lesson. And I just put a note about that in here. Okay, and the last ordination that I'm showing here is a non-metric dimensional scaling. So PCOA, in addition to the PCOA name, is also called metric dimensional scaling. Non-metric is kind of like the non-parametric version of this. So instead of using actual distances, it uses ranked distances. And the other difference about non-metric dimensional scaling is that it's not using, so instead of rotating your axes around your data, you're actually formatting the data to fit into two axes. So because it's ranked, our axes don't explain a percent variability, right? Because it's a non-parametric method, so we don't know what the actual distances are. So the distance between 2 points is not actually meaningful in terms of absolute distance or absolute differences. But so let me just run this, I guess. So ordinate, we're still using the same function from phyloseq ordinate. Now the method is NMDS and the distance is Bray. So we see similar results, right? Biofilm and water separate out from the rock samples, and we have some separation among the rock samples. A difference in interpretation here is, again, this is adjusting the data to fit into two axes rather than adjusting axes to fit the data. So it's an iterative process. And so each time you run this, it's actually going to find a different unique solution. So if you look at this first table of results here, it runs 20 iterations until it decides on a stress number. And this stress is basically how much you had to change the distance layout in the data in order to fit into these axes. So you don't want a very high stress in this analysis. And I think the recommendation is usually you want a stress that's less than 0.2. In this case, in our 20th iteration, we got a stress that is essentially 0.05. So that's pretty good. So an MDS in this case would be a good approach as well. So I think that's all I wanted to say. I know that was a lot. I guess I'll take questions.
That's awesome, Liz. Thank you. I have two quick questions for you.
Sure.
So the stress that you last showed was really interesting to look at. And is that one of the primary way that you decide on the method and the distance for the ordination?
Yeah, good question.
I know that's like a kind of a loaded question in some cases, but
yeah. So sometimes it's pretty straightforward. Like if your PCOA has pretty low variability explained, and then the stress is decent, and the NMDS, you would just go with the NMDS. In this example, all three methods actually worked out pretty okay. So it's not clear which one you should use, in which case I think you should account for all of them. Like obviously you can't put all of them in a publication, but you could say something that in the publication that you tried all three and the interpretation was not too different, but we explored this difference and it showed us this, So I think in this case, because all of them are decent, you would have to do something like
that. Yeah. Okay. Ella or Brittany, do you guys have any other comments about the stress? I know there was one question about colors, but.
Yeah, what I've heard in the past is that you want the stress to be under 0.1. Say
that again, Ella?
Sorry. So what I heard in the past is that you kind of want your stress to be under 0.1.
Yeah.
That's the value that I usually hear.
I've seen both. So again, I don't think it's like do you want a P less than 0.1 or a P less than 0.05? It's kind of like a open question. Right. There's no right number. But yeah, I have seen 0.1 also.
And then one of the other questions that came up was about the color changing. So I know that you assigned the color for the substrate here, but can you also, like those are like obviously the like default ggplot colors. Can you also do like the color brewer and like add in the palette for that pretty easily in these plot ordination things? And then if I understand, I've used PhyloSeq, but not 100% of the time. But can you pretty much put all the ggplot things into this?
Yeah, so I think that PhyloSeq is built on top of ggplot. Yeah. So like this is a PhyloSeq function, but it's taking what looks like the ggplot syntax, right? Like you can add geom points on top of it. So yeah, I think any modifications that you could do to a ggplot, you could do to a PhyloSeq plot.
Yeah. So you can like add the scale manual values and add in the colors and stuff. Yeah. Cool.
All that and the color brewer and whatever.
Mm-hmm. And then Brittany had one other question about similarly about ggtree and just like overall using ggplot versus ggtree. I think that is just an add-on to ggplot. Like I've used ggtree and oh my god, another one. I completely am blanking on it, but another GG something, and then one that was also GG. Oh my god, I'm going blanking. to make a 3G ternary, to make a ternary plot, like a triangular plot with 3 axes. So there's a lot of like add-ons to ggplots. I'm pretty sure that's what ggtree is.
I have never used it, so I don't know. Yeah. But there's a lot of ways of doing all of these things. I think you and I were discussing before. So, I mean, if you find a way that works for you and you prefer it, that's fine.
Yeah, I mean, right, like the other thing is that with some of the ways that PhyloSeq is written, especially how you can add on these things, it's really easy to follow Google online tutorials because a lot of people using them and like interlocking the different packages. And so there ends up being a little bit of a community, which is nice.
Yeah, agree. There's a million PhyloSeq tutorials that are really, really helpful. And I essentially, like, I patch all of the time. Every time I use PhyloSeq, I kind of patch it together based on all of these other tutorials that I've found.
Mm-hmm. For sure. Awesome. Anything else we should cover?
I think that was a lot. That's like a half hour, so.
No, that was awesome though.
Okay. Great. Well, I will get this posted as soon as possible.
Cool. Thank you for going over that. And it was great.
Thanks, Liz.
Bye.